Hello everyone. In the last session we were studying about the sex determination and the types of sex determination. There are 4 types of sex determination: XX, XY, XSX0, ZZZW and ZZZO. So we have seen the examples for all those types of sex determination. So with that we also studied about the sex determination in humans, how many pairs of chromosomes, autosomes are there, how many pairs of sex chromosomes are there. So we all done with all those sex determination types and sex determination in humans. We'll proceed on to the next concept. The next concept is sex determination in honeybee. In honeybees, how many pairs of chromosomes are there in honeybee, how it fertilizes, in case of fertilization what type of honeybee it produces. So we'll see all those one by one. So we are in the same concept, sex determination, but we are calculating or we are observing the sex determination in honeybees. So the female honeybee, concentrate this, if the female honeybee, when it fertilizes, in case of fertilization, I'm speaking here fertilization means if the female honeybee fertilizes with the male honeybee, it reproduces or it gives rise to the offspring which is a female one, or we are going to call that as queens or worker honeybee in case of fertilization. So once if the female fertilizes with the 15 to 20 male honeybee, the resultant offspring will be queens or workers. Okay, only queens or workers or female honeybee. If the female honeybee, if it do not fertilizes with any male honeybee, it directly produces a honeybee. Those honeybee will be a male. That male honeybees we are going to call that as drones. How the female honeybee without fertilization it produces the drones or it produces the male honeybee. That process it is known as, the process is called as Parthenogenesis. By means of Parthenogenesis, by means of that process, the female bee without fertilization by means of mitosis it produces male honeybee. In the it literally means drones, and female honeybee, this is the resultant offspring for it came across by means of fertilization. So we'll see the chromosome number. The female honeybees, these are having 32 chromosomes. This is the diploid set. Okay, if it is crossing with the male honeybee, it give rise to what type of honeybee it is giving rise to? It is giving rise to queens or workers. So that queens or workers will be having 32 set of or 32 chromosomes. If the same honeybee, if it do not undergoing fertilization, if it is not mating with the male drone, in that time or in that case, it give rise to the drones or male honeybee. So you might have, you'll have a confusion here. So 32, so this is the female honeybee chromosome number. Okay, so it will be having some procedures, like first it undergoes meiosis, there it produces gametes, those gametes will be having 16 number of chromosomes. When it pairs with or when it mate with the male male honeybee, those honeybees will be producing or that secrete sperms. Those sperms will be having a set of chromosomes. The resultant product, what we are getting here, we are getting in the F1 generation, we are getting the queens or workers, but the resultant product will be 32 chromosomes. How we got the 32 chromosomes? When we add 16 and 16, we'll get 32 chromosomes, but the sex of the honeybee will be a female one, or we are calling this as queens or workers. But here, without meiosis or without fertilization, it is undergoing, first it will reproduces, first it will divide. Later, the 32 chromosomes honeybee, by means of parthenogenesis, it is by means of only mitosis, it produces male honeybee or drones. So that's all about the sex determination in honeybee. So we'll move forward with the next concept, that is mutation. Mutation. What is mutation? So mutation, it is a phenomena. It is a process where the alteration of genetic material, the alteration of genes or the alteration of DNA sequences takes place. With the result of that phenomena, the genotype and phenotype, these two alters, these two undergo some changes occurs in the genotype and phenotype. The whole process or the whole phenomena is known as mutation. Here the DNA sequence, it will just alter, or what we studied in the class 11, the DNA sequence or the DNA strand, one strand and one more strand, base pairs, all these we have completed in class 11 itself, right? If something get altered in the sequences or in the base pairs, that results in the changes in genotype and phenotype in an organism. If an organism, he has undergone some changes in his DNA sequences, those DNA sequences results in the change or alteration in its or his genotype and phenotype. Still we are not getting what is mutation. Still we can't make out what is genotype. That's all about the definition. What I told you, that's all about the definition. By definition we can't make out what is mutation. So we'll have an example here. We'll move on to the next concept. First we'll understand how the mutation occurs. With that I'll give one example and then we'll move on to the next. So how it forms, why the gene or the DNA sequences it get altered. So alteration means some of the DNA sequences it get deleted, some of the DNA sequences it get added, maybe either increasing or decreasing. Both the possibilities can occur in the mutation. In the case of decreasing or in the case of deleting of DNA sequences, what will be the result of that DNA strand, how it will become. So we'll see the thing first, on what basis or how the mutation causes. The factors I'm writing here. some environmental factors like chemical factor or radiation factor results in the alteration of DNA sequences. Okay, so like for example, to this DNA sequence, if we pass the UV photon, okay, so earlier the DNA was in a proper manner, all right. When we pass the UV rays to this DNA sequence, some changes occur in the DNA sequence. What are those changes? I'll draw one diagram here representing the mutation or the alteration in DNA sequences. So how will be the, what is the diagram? So you can observe here. See, some misprint, mismatch in the DNA sequences, nucleotide sequences. So that is nothing but mutation. Got it? So is this the longer, it will stay for a longer period? Yes, it will stay for a longer period. So the environmental factors or the mutation, it brings significance in some individuals, but in some individuals it will be harmful. It shows an harmful effect in the individuals. Okay, so the environmental will come to the mutation. We have seen the definition and we have referred the factors also. Literally we came to know how the mutation forms. These mutations are of two types. One is gene mutation or point mutation, and one more is chromosomal mutation. So there are two types. In your book, NCERT book, they have given the point mutation or gene mutation, both are correct. And one more they have given in your book is frame shift mutation. We'll speak about the frame shift mutation in chapter number 6. But for your reference, I am explaining about the chromosomal mutation. But this is more than enough. Refer this, learn this gene mutation or point mutation. This is also very easy. No need of extra concentrating or it is not much difficult. Chromosomal mutation. So online definition I am giving here. So gene mutation, it is confined to gene which results in the alteration of nucleotide sequences. Here, what is gene? Gene is the fragment of DNA, right? So I am coming back to the same diagram, but only to describe you, only to explain about the gene or point mutation. So it is confined to gene. Gene mutation is confined to gene. It results in the alteration of what? Genes, or alteration of DNA nucleotide, polynucleotide sequences. That alters the, confined to gene, that alters the sequences, polynucleotide sequences in DNA strand. So that results, or that is known as gene mutation or point mutation. Here one example is there. The example is sickle cell anemia, which beautifully explains about the gene mutation or point mutation. What is sickle cell anemia? One more example is there. I am going to give you in chromosomal mutation. So these two explanation comes under the genetic disorders. that I'll tell you in the next session. So keep this as it is. Sickle cell anemia, it has to be in your mind. When I start telling about the sickle cell anemia, you have to remember, yes, this comes under the gene mutation, only a small or only in the gene. If the alteration occur in the gene, that too, what gene, that too, in what form the alteration is taking place, that too in the polynucleotide sequence. If something happens, some alteration occurs, that directly hit to the gene, that directly changes the formation of gene. Okay, so if one gene, if it get changed, if it get altered in its position, or if it get alters in its manners, the phenotype or genotype, it get alters. The phenotype and genotype of the individual, it get alters. So results in the formation of sickle cell anemia. Yes, that is the disorder. Yes, that is the disease carried by so many human beings. Okay, someone are suffering from this type of disease. Why? Because they are suffering from gene mutation or point mutation. Some alteration has happened in their RBC or hemoglobin. Because of that, they are suffering from sickle cell anemia. This I'll explain you in detail in the next session when I come to the genetic disorders. So only alteration in nucleotide sequences known as gene mutation. What about chromosomal mutation? Consider we are talking about the sex chromosome. We have one X chromosome. Okay, I'll write only XX, no need of drawing the chromosomes and all. Consider we are talking about the XX and XY type of chromosomes here. What happens if in females one X chromosome get deleted due to some mutations, due to some environmental factors, or due to some misprint or mismatch in cell division? The X chromosomes completely get deleted. Female, she is carrying only 44A and X type of chromosome. So what do you call those type of females? Those females will be or will become sterile. So what is sterility? As you know, she can't give birth to young one. So one more, what I told here I am talking about the deletion of chromosome. What happened in males? In males, what happened if one chromosome, X chromosome, added? So what the males are called as? Those are called as, or they will be suffering from one syndrome known as Klinefelter's syndrome. What is that? So that is a syndrome caused by the adding of one more extra X chromosome. So all these things, one is sickle cell anemia, phenylketonuria, Turner syndrome, Klinefelter syndrome, everything I am going to explain you in the next session. Okay, so keep this in mind. The chromosomal mutation is nothing but increasing or decreasing of chromosome in an individual. It leads to chromosomal mutation. The factors are because of the chemical or radiation factor or environmental factor or misprint or mismatch during cell division. So many reasons we can give you, but you have to remember gene mutation or point mutation. One more is frame mutation, that I am going to tell you in chapter number 6. Only for your reference, only to refer the genetic disorders or chromosomal disorders. Keep this in mind. Okay, so so that's all about the according to Dobzhansky, the mutation is known as misprint or mismatch in cell division according to Dobzhansky. Okay, so what left? Yeah, one more thing I should tell here. This alteration in DNA sequences leads to chromosomal aberration. Chromosomal aberration. So what is aberration? Alteration or aberration. So this type of chromosomal aberration seen in cancer cells. I think you got my point here. The cancer cells, these will be having some chromosomal alteration, some chromosomal disturbances, leads to what is that? Some kind of diseases or some kind of cancer type of diseases. Okay, so what you have to what you have to remember in the mutation, from the mutation evolution takes place, and that evolution is the plus point in organism. But in the mutation, in this concept, we are not talking about the significance of mutation, but we are concentrating only on the disorders and diseases or syndromes. What will happen if the mutation occurs in human body? Okay, only that we are understanding in the in this concept. Okay, so mutation is alteration in DNA sequences or DNA polynucleotide sequences that alternates the genotype and phenotype of an individual. In literally we can say misprint or mismatch in cell division is known as mutation, but don't follow that definition. It was told by Dobzhansky. He was the scientist. Okay, so all these things you have to refer from mutation.